So hi everyone, welcome to class number 7 of systems biology. It's a good time if you feel like it, to do our little breathing ritual. So if you feel like it, you can join me in taking a nice deep sigh of relief. Which is something I think really recommend you try when you're sitting on your computer or stressed out. If you remember to breathe, you remember yourself somehow. Summer's kind of suddenly began. A few months ago, we planted passiflora, passion fruit tree plant, and it's unbelievable how it grew to this a size like this in just a few months, and now it gave fruit for the first time this week, which is a very beautiful thing to see. It makes me think of the topic of this class. Just like last time, we'll talk about how a seed becomes a plant, or a fertilized egg becomes an embryo, the process of pattern formation, how it happens. So let's take another nice deep sigh of relief. How it happens so reproducibly, so accurately. And another nice deep sigh of relief. Thank you. Yeah, you might ask why when people come in late, I do that because otherwise, when somebody comes in, everybody notices it, and if we don't, it creates a little bit of tension, like tiny little bit of tension, and just by acknowledging it, it's very welcoming. It's okay to be late, it doesn't matter. So I will start by reviewing a little bit what we did last time with the exponential gradients, and then today we'll talk about a very different mechanism to achieve a similar robustness. It has more biological details. And okay. So the question we're asking is, we're showing kind of mechanisms that can generate robust patterns despite variations in protein you know what we have to do? We have to take a nice deep sigh of relief in your honor. Production rates. Good. So we talked about this amazing phenomenon where an egg becomes it starts dividing, 2 cells, 4 cells, 8 cells, 16 cells. All cells have the same DNA. How is it that one cell becomes a muscle cell, another cell becomes a skin cell? It has to do with expressing different proteins. The proteins of a muscle cell are different from proteins of a brain cell, etc. How is this done? This field called developmental biology asks the question how patterns are formed from an unpatterned source. And there are many different kinds of mechanisms, but one that's primarily has been best studied has to do with morphogens, which are molecules that are produced somewhere, and then they move, and they create gradients. And the cells have receptors. Each cell has thousands of receptors on its surface to sense the morphogen that's flowing over it, and that way you can detect the concentration of the morphogen. So if you're close to the source, the concentration is high. If you're far away from the source, the concentration is low, and that way the cells can have a sense of where they are. Yeah?
All the cells are exactly the same, how these morphogens are created only one point and not everywhere.
So the question is the chicken and an egg problem. So how does the initial source pattern determined? Let's take a nice deep sigh of relief. And so of course there's an initial pattern breaking in symmetry that can come from the mother. For example, in the fruit fly, Drosophila, the mother deposits, because the mother is not symmetrical, has head tilted, deposits RNA of a transcription factor called bicoid at one end of the egg, and that the gradient formed, it determines the head-tail axis of the Drosophila. Once there's a coarse pattern, let's say several parts according to the concentration of the early morphogen, each part can now become special, and let's say the middle part secretes the second morphogen that now patterns the third region. So there's a hierarchy, temporal sequence of morphogens for finer and finer patterns. In the frog egg, it's the sperm entry point that determines the position of the initial asymmetry, for example. So there's something that breaks the initial symmetry. Again, in most cases, the answer isn't known, but there are some cases where the answer is, we think we know.
Morphogens are used only for this patterning, or do they have some other biological purpose?
Do morphogens exist only for the purpose of patterning, or do they have other uses? As far as I know, they're specific for patterning, but I could be just, I don't know. It could be that they have other. The same, it's interesting, the evolution preserves. So the morphogen I'll talk about today, BMP, this in fruit flies, is also operating in us to make hands, for example. So the same molecules, very similar ones, make patterns in different organisms, but what they do make is quite dramatically different. But the principles are quite similar, apparently. Even the molecules are preserved that determine the positional information. So last time we discussed, just as a review, we discussed a situation where the morphogen is produced at one position. Morphogen is produced and diffuses. So x equals 0. This is the position. And morphogen is produced at x equals 0, diffuses with a diffusion constant d, and is degraded. And we compared two models, one where the morphogen diffuses and is degraded with regular linear kinetics, and we saw that this leads to an exponential gradient and that when I say robustness, what I mean is that if we change the production rate of morphogen, so we have a different, let's say we make less of it, then robustness means that the pattern doesn't change significantly. That's what robustness means. And you remember that the mechanism of patterning is that there's a threshold, and this determines the position of where you change from sulfate A to sulfate B. It's determined by when the morphogen crosses the thresholds. The cells here have receptors on their surface that can detect this diffusing morphogen. So the cells far away see just a few, right? Because it diffuses and it gets degraded. And for this kind of process, we saw that this is not robust because changing the initial concentration changes the profile. And in this case, you never actually cross the threshold, and you don't have cell type A at all. And we saw that a different model I'm just reviewing what we did last time. With self-enhanced degradation has a very different kind of shape. It looks like something over x squared. And if you change the initial levels, apart from a small region where the profile isn't robust, you get exactly the same shape, or almost exactly the same shape. It depends very, very weakly on the initial conditions. and this is robust. Bigger letters. Yeah. So, any questions about this?
Could you say what is the mechanism that the cells use to detect between the different thresholds? How can they detect multiple thresholds?
Thanks. So how do cells detect the multiple thresholds? In the morphogens, I think some people define morphogens as making at least 3 different regions, which means at least 2 different thresholds. In general, there's receptors on the cell surface, like I said, that detect the morphogen, they bind it, and they start a signaling cascade. This is just like what we talked about in bacterial chemotaxis, where the receptor senses the attractant and affects the rate of phosphorylation of a protein that diffuses inside the cell. The same idea, there's phosphorylation of proteins, and they go into the nucleus of the cell, and sometimes there's a whole cascade of phosphorylations, and then they phosphorylate, let's say, a transcription factor that binds to the DNA and causes genes to be expressed. So to make the specific protein. So in general, it's a signaling cascade that converts the external concentration to turning on specific genes. And you also might remember that you can have here transcriptional networks, so that you can have proteins like a protein A that activates protein B, and protein B activates protein A. And this creates a memory. So this can mean this cell, even after the morphogen is gone, remembers now that it is, let's say, a muscle cell. So we talked about this in memory circuits, if you remember, in one of the classes. So all the things we talk about in the course kind of progressively are supposed to build for people who are new to biology and understanding how signals come from the outside of the cell to receptors, to this phosphorylation cascades or whatever signaling networks, and then to transcription. And transcription can make transcription circuits that have memory and delays and all this. So once you have receptors, the mechanisms for threshold detection can be quite diverse. But for example, if you have a lot of morphogen at a certain point, you can have a lot of this phosphorylation, etc. And then the concentration of this transcription factor is high, and it can express many kinds of genes that have its binding site. But in other cells where the concentration is low, there's less of this, so it only binds to the strongest binding sites. And this difference can create a difference between the sets of genes expressed in the two areas. And there you have an intuitive explanation. But I can't tell you what is it that makes the threshold very sharp. There's a lot of additional circuitry inside the cell that can do that. And
how can they detect like 2 thresholds? I can understand that the sigmoidal curve could give you low and high concentration. And how can they know now I'm in the middle of 2 thresholds?
One easy way has to do with the binding sites. So if you have binding sites for this transcription factor that are only work when you have a high concentration of transcription factor, others that work when you have a medium concentration or high, and others that work only when you have a low concentration. That way you can have 3 kinds of responses, and that's tuned in the DNA sequence. But that's only partial. There's additional circuits here that can give you the computation that you need. Now, these are very general mechanisms. I want to turn now to look in more detail at a very specific event in development of the fruit fly, so you can get more of a flavor of the way the biology feels like. And I think the main point in this whole section of classes where we talk about robustness, the principle of robustness, is that robustness can help us find the mechanisms that work in the cell out of many possible mechanisms that only work on paper. I say this kind of as a joke, but for a given task like pattern formation, you can write many different mechanisms, but when you also want them to be robust, it really eliminates a vast majority, we'll see today how you can do that again, of possible mechanisms, and you end up with very special mechanisms, and they make predictions, experimental predictions that you can again test experimentally and help us explain some of the biological complexity that we see. All right, so we'll talk about work by Nama Barkai and Avigdor Eldar and Benny Shiloh that work here at Weizmann that worked on a particular well-studied case of developmental patterning using one of the best studied organisms, the fruit fly Drosophila. It's nice because it has a short generation time, and it's been studied for about 100 years. A lot of information is known, etc. And the fruit fly starts from an egg, and this egg starts dividing very, very rapidly. In fact, just the nuclei divide. And after 2.5 hours, okay, so we're here. After 2.5 hours, the egg looks like a cylinder that's about a few hundred microns across, and all the cells are on the layer on the outside of this cylinder. And if we look at the cross section of the cylinder, so the cells are here, and there's a layer here of water, basically, where morphogens and stuff can flow. This perivitellin layer. So the morphogens are flowing on top of the cells. And what we're going to concentrate on is patterning. So there were earlier morphogen events, so we have a coarse pattern already in place. And we're going to concentrate on patterning of this dorsal region here. Our goal will be to divide this region into smaller regions. And the network that does this, which you can define by mutations. Mutations in 3 different genes can disrupt this particular patterning event. It's a network of 3 proteins that cooperate to do this. Right, and these 3 proteins are M, the morphogen, which is called BMP, like I mentioned. There is an inhibitor. So this protein I can bind M, and when it binds M can't talk to the receptors. And it's a pretty tight binding. So when it binds, it doesn't let go. And there's a protein protease. Protease is an enzyme that can cut proteins. And this, it's like a scissors. It can cut proteins. and it cuts I. It can also cut I when it's inside, is bound when it's bound with the morphogen to release the morphogen. Okay? So these three proteins. They're called BMP, SOG, and toloid, the names of these proteins. So in our system here, the morphogen doesn't get degraded, unlike what we did in last time, where the morphogen degradation was critical for us. Here, it doesn't get degraded. So we need a different mechanism.
It gets degraded along the z-axis?
Does it get degraded along the z-axis? No. And in fact, we're going to assume here that this is like an infinite cylinder, where in fact it's not infinite, it's finite. But it's kind of a quasi-one, it's a good assumption, quasi-one-dimensional process. And we're just going to take this coordinate here. It'll be actually, we're going to use just linear coordinates. Yeah. We're going to even ignore the curvature. It's not important in this. I just want to say the length of this is 50 cells, more or less. So 50 cell width. And they are identical in the beginning of this process, and we want to divide them up into sections. Okay. Now, I want to tell you where these proteins are. Yeah.
Which length? 50 cells?
Yeah, so this is... Yeah. Okay.
Another question. The timing of the morphogen production, should it be very precise in the development stage? Because the 50 cells is very accurate timing in the development of the...
Yeah. The question is, I talk robustness to errors in timing. Let's say it's half an hour late.
Yeah.
So that could be quite disastrous. Timing also needs to be
Very robust.
quite precise, yeah. Especially things move very quickly in the fruit fly embryo at these early stages.
There is some mechanism that ignite.
Yeah, there is mechanisms. The patterning of this region makes regions that when they're committed to become their cell type, they start secreting. So these steps are tied together. But I don't know too much about mechanisms for robust timing. So people here, several people asked me to talk about unknown things we don't understand. It's like, that's a great question. Also, when you think about how there are many different clocks here working in parallel. So there's not only, while this is patterned, this is patterned, this is patterned, this is patterned. So these clocks need to work kind of in a synchronized way. Otherwise, you have quite disastrous. So mistakes in the early pattern formation are very disastrous. I mean, you can have the whole embryo. You know, if you don't have a head or you don't have a nervous system, or it's like major catastrophes. And those catastrophes are very rare. So you can play around with the embryo, change temperature. For example, if you change temperature from 25 degrees to 18 degrees, the whole development is slower. Let's say it takes 2 times slower. But still you get a perfect fly. So that's all these things are like, there's a lot, as I say, there's a lot more to understand here. Yeah.
What do the mutants look like for these three?
Well, I guess without a morphogen you don't get patterning. And without inhibitor, you get a kind of a hyperactive morphogen. So it's like the pattern is as if the morphogen was high everywhere. And without protease, it's like you have too much inhibitor, which means no it's like no morphogen, something like that. So you can see in the embryo some defects. I just want to tell you where these are, where they are. Okay, let's. Did I answer? Wait, I don't know. Did I answer your question? Or?
I was curious about the names, I guess, because sometimes they tell you what the...
Oh, yeah, I don't know, but BMP is bone morphogen factor because it was discovered first in bones, and then the very close molecules found in fruit flies, etc. So, but I don't know about these others. Maybe other people here know. I don't know. There's people who works on flies here. No one? There's like 3 labs in Weizmann. Okay. So where are they? The inhibitor. It's what's important for you to know is that inhibitor is produced in these regions here. So the inhibitor is made here. Made in the regions next to our region. And morphogen is present initially everywhere. It's not. It's kind of. It has a constant level everywhere. So morphogen is everywhere, and the protease is uniform here in the DR. Again, also constant level. All right. So what's the deal here? What's the purpose of the circuit? So I'm going to draw here. This will be the position. So this dorsal region, I'm kind of making it a line going along the dorsal region. And the goal, the way the patterning works is that so you see the morphogen can be either free, which means it's active, or inhibited, right? The morphogen can be, so the way this works is that free or active morphogen, for the patterning to work, it should be in the middle. I didn't draw this very symmetrically, but in the middle, it should be high. In the end of the process, we're looking for something. In the middle, it should be high, and then go down.
Only in this chart is axis is position along the circle.
Yeah, it's. So this is x. So the morphogen needs to be high here in the middle, and low towards the ends. So if there's a threshold here, there's one. The patterning should have one section here A, another section here B, and another section here C. That's the way this region is patterned. Or the goal of this is free M, which is high at the midline. And I just want to tell you that you can measure this activity profile. How can you measure this activity profile? You take the cells and you hook up the signaling pathway that detects the morphogen. You hook it up so that when it's active, it makes a green fluorescent protein, for example, the protein I told you about from jellyfish. So the cells where that sense the active morphogen at high concentrations are very green, and ones that at low concentrations are less green, etc. And so you can look at the green profile and understand the morphogen activity profile by making genetically engineered flies who become green in a morphogen responsive promoter. So this technology exists. You can make fruit flies basically play with their genome at will almost. You can make them grow feet instead of antenna, etc. All these things are standard. Yeah.
The morphogen is present everywhere. Why are we looking just at the DR region? What's happening if we go far away, for example, to the other side? To the opposite
Okay, so if there's morphogen here, then the cell there's a pre-pattern already in place at 2.5 hours. So these cells here don't care about the morphogen. They don't have the receptors. These cells are special. They have the receptor. They care about it. But they're identical except for caring about the morphogen. So this step in patterning is about the relative levels of morphogen. Thanks for the question. I want to tell you that experiments show that reducing P, I, or M by 50% doesn't change the M of X activity profile at the end of the process. The way you do this is by deleting one of the two copies of each of these genes on the two chromosomes. Animals have two chromosomes for each gene, from mama and from papa. So you delete, let's say, mama, and you measure, and indeed, with one gene, they make 50% less of the protein. Or you can take and add an extra copy of the gene, so you make 50% more. And you get the same profile. Okay? So this is what's called robustness. Of course, if I take away both copies of the gene, so I have no morphogen, no pattern. But if I take away 50%, it can tolerate it. Not only tolerate it, it looks perfectly the same. So we need to explain that robustness. And we can't use the mechanism of last time because we have no degradation of the morphogen. So we need something else.
You delete one gene at a time?
Yeah. Are you asking what happens if we delete all three? Good question. According to the model, it's supposed to work. All right, so let's start with this information. Let's start to look at it. And I'm going to start with two models. One's going to be not robust. And then we're going to use the robustness principle, do a kind of numerical scan of parameters, zero in on models that are robust, see what they look like, see that you can solve them analytically, see the solution, try to understand what's going on. Okay? Biochemical predictions. Yeah. So we start with, so we look at dosage experiments, we defined our players, robustness. And now the first thing you do as a modeler, or not only as a modeler, all biologists use models. Usually we use models which are prose models, that is to say words. And they're very powerful because you can, they generate all these experiments. I mean, we're done basically without math models at all, and you can discover a lot. But when you talk about things like robustness, you need the math, I think, because it's kind of it has more subtlety, and that's so the prose model goes as follows. It's easy to understand. The inhibitor is made at the sides. So the inhibitor concentration, I'm going to make it draw it like this. The inhibitor concentration is high here at the sides, and it diffuses and gets degraded. You remember the inhibitor diffuses and gets degraded because it has this protease that degrades it. So it's made at the sides. This is just like last time. It diffuses into, and where its concentration is high, it can bind the morphogen and inactivate it. So the free morphogen should be high where the inhibitor is low, right? So that's the way it works. So I, diffuses and it gets degraded, and this causes this exponential gradient that decays from both sides. The problem is it's not robust to changes in I, because if we lower I by 50%, we get profiles that look like this, and we get morphogens patterns that look like this, and the profile changes. In everything, I, M, P, changing any of the parameters affects the profile. There's no. So that doesn't work. So what Naaman and Victor did was to write down a diffusion model for all the proteins in the circuit. And this model results in a set of 3 reaction diffusion equations with a lot of parameters, like diffusion constants, binding constants, degradation rates. And so they wrote a model, and then they simulated the model, and they varied the parameters. For instance, the diffusion constant of the morphogen by, let's say, 4 orders of magnitude. All the parameters, right? The degradation rate. So they sampled this. They told the computer some values for the parameters, then they solved those equations on the computer, and they looked at the morphogen profile. And then, with the same parameters, they tested for robustness. They varied the concentration of the proteins by 50% and asked, does the profile change? And then they searched, is there a region of parameter space where you do get robustness? Right? So let's see what happened. So I'm going to write down now the equations. Instead of 1 equation that we did last time, it will be 3 equations. So 3 times more equations. That's okay. And we're going to solve them, I think. So let's start with, what should we start with, actually? Start with the inhibitor. So the rate of change of an inhibitor. So it diffuses, like we said, the inhibitor diffuses. So this is the diffusion equation, like we did last time, the diffusion constant times the second derivative with respect to space. So this is diffusion. And now I want to describe this process where the morphogen and the inhibitor bind each other. And that has a rate k. That's I and m meet each other according to their concentrations. And the rate at which they form a complex that doesn't come apart is k. So this I goes into the complex at a rate k times the concentration of I times the concentration of m. So this goes into a complex. We'll call this complex C. That's the complex of m and I together. This is the rate k. And this is mass action. So you multiply the concentration of I times the concentration of free morphogen times this rate constant. And then, of course, we lose a free I. It goes into the C, into the complex C. So see, this is the complex of M and I. And I can also be degraded. By our protease at a rate alpha. So we have here an equation for diffusion, complex formation, and degradation. Any questions about this?
Where do you add the fact that you don't have them in the DR?
That has to do with boundary conditions. So at t equals 0, I is 0, except for these boundaries where that has to do with boundary conditions. Thanks. And we also need an equation for the complex, which can diffuse. It's formed when I and M bind. So every loss of I is actually a gain of a complex. It's formed here by the same collision. And it gets degraded. And you remember that the protease can cut the complex and cut the I, and that liberates an M. So the complex can be degraded, and the free morphogen can diffuse. It has its own diffusion constant. It's also lost when a complex is formed. When a complex is formed, one I is lost, one M is lost, and one C is gained. And it gets, and this is interesting, when a complex is degraded, a free M is gained. So the degradation of a complex equals the gain of one free M. You see that? So that's the way you describe this biochemistry. Yeah, as I said in the beginning, in this system, the morphogen isn't degraded. Its level is constant. That's right. So in these equations, I added a lot of biochemistry known from the system, of course. The inhibitor is degraded either when it's free or when it's in a complex. So the degradation of a complex actually is also a loss of an inhibitor because it went through the complex through here, it gets degraded. Yeah? Did I explain myself?
It's still, I don't understand. Is there, because when you lose a complex, you lose an I, is there a relationship between alpha I and alpha C that we haven't expressed fully?
Yeah, so the question is, when you lose a complex, you lose an I. It's correct. But you can still have a different degradation rate for a free I and for an I in the complex, and they don't have to have a relationship. They can biochemically be anything, basically. These diffusion constants could be anything because molecules can be sticky in biology. And they can actually be even not move if they're sticky. And if they're not sticky, they can move with, let's say, a protein can move, let's say, with 100 micron square per second diffusion constant or something. So there's a whole range you can play with that.
P is saturated?
And P it's not saturated, but it's constant throughout. So I wrote here P just to, this is like a constant level throughout. The problem, it doesn't move. It's just around. It's constant. It's constant. It's a parameter. But if we want to test robustness for it by solving these equations with one level of P and then taking 50% of that level and seeing how that affects the profile. And by the way, these equations are too difficult as far as I know to solve analytically. You can try. They're nonlinear equations. And you can solve them analytically. Maybe you can, but I can't. And you can solve them on the computer. You give initial conditions, i's at the boundary, and you let the computer propagate the diffusion equation. It reaches a steady state. You get a profile. The profile stabilizes, and you get something high at the midline. And then you can take the amount of P, the amount of M, the amount of I that you started with in the initial conditions, change them by 50%, run the simulation again, and get a profile, and test how close is the perturbed profile to the original profile. And according to the criteria they use, let's say that the profile changes by less than 5% or something. 97%. 97%, of course, of the choices of our parameters, dm, di, blah, 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 alpha c. So you can have robustness, and we're going to take a break now. מוזיקה שוב פעם? תודה. Okay, so we looked at these equations. There were some questions in the break that I was asked why I write this and not a Michaelis-Menten term. So Owen sent, I think, all of you Appendix A from the book that shows how you can get Michaelis-Menten terms from mass action kinetics. So this is the basic kinetics. The rate of change has to do with k on and k off terms. And if you take them in steady state, under certain conditions, you get to Michaelis-Menten forms. But this is the more basic dynamics. And you can read that in the Appendix A of the book. here I added the K, which is the binding, but I didn't take the K off, which is unbinding, because we know that this complex lasts for a longer time than the lifetime of this system. So we don't need to worry about that reaction. But when the protease cuts I, that's the way the complex goes away. And I was asked about morphogen. It's not degraded. How come it doesn't accumulate to infinity? That's because it's also not made. In the time of this system, it was made before. It's diffusing all around the fluid layer. It's not made, not degraded. It's basically in a constant total level in this process. What gets redistributed is is the concentration of the free morphogen. So, I was also asked, what about the 2.5%? Right. So if you want to know, you should read the paper. But let's say they're in kind of in the gray zone. But let's say 99.5% are not robust according to the threshold, and 0.5% are. So less than 1% of in the parameter space of this model of the parameters is robust. And now they took a look. Who are those 0.5%? Let's take a nice deep sigh of relief. And it turns out that they all are basically the same model that was sampled very closely many times. And the robust models have two things in common. The morphogen, the free morphogen, doesn't diffuse. I mean, in all the robust models, the diffusion constant of the free morphogen is very small compared to all the other diffusion constants. So that means that the free morphogen is kind of sticky. It doesn't diffuse. It only diffuses when it's bound to the inhibitor. So the inhibitor that shuts it down is also what allows it to move. Okay? So free M doesn't move, doesn't diffuse. Only moves when bound to I. So I is actually acting like a taxi, not only an inhibitor, it's like a taxi, or random walking taxi. And the second thing is that alpha I was very small. So free inhibitor doesn't get degraded. Only the inhibitor in the taxi, in this complex, gets degraded by the scissors. So those are two biochemical predictions, you can say. If you want robustness, you need these two weird features. And I'll try to explain why those two features give you a robustness now. But they're definitely not something you might have thought about. And they are also things that you can experimentally test, right? Just you need to know to look for these kind of things when you experimentally test. So in fact, what this means is that you get the same set of equations, but for the robust models, this doesn't exist. This is 0. Or in other words, you can neglect this term, the diffusion, dm is 0, and you can neglect this term. So you get a little bit simpler set of equations. And it turns out that this set of equations you can solve analytically. The steady state profiles. You can solve them, and we're going to solve them right now. You can solve it on the blackboard. And see for yourself mathematically whether the profile depends on all the parameters that are left. There's still, diffusion constants, protein levels, degradation rates, etc.
You solve analytically for the steady state? Or?
Yeah, steady state. Mm-hmm. And I think also not for the steady state, but that's not so important for us now. So we're going to solve it for the steady state. Are you ready?
Yes.
Okay, hold on tight. So steady state means that all the time derivatives are 0, right? Nothing's changing anymore. That's what steady state means. How are we going to solve this? Yeah, let's try. I'm going to try to solve it in this little area here. Can we do it? Okay. So we're going to call these equations 1, 2, and 3. So we'll start with adding equation 2 plus 3. What happens when we add equations 2 plus 3? You see that every complex formed is one morphogen lost, so these terms cancel out. And every complex degraded is one morphogen gained. So these terms cancel out when we add. And we're left with the profile, the profile of the complex is something whose second derivative is 0. No? So what functions their second derivative is 0? A line. Because you see, if I take the first derivative, I have a, and second derivative is 0. Now, because of symmetry, see, this line the complex can either look like this or like this. But because of symmetry, because the right and the left are the same, we can't have a slope. So we arrive at the conclusion that the complex is constant in space. So that's interesting. So the concentration of the complex is flat. It's constant in space. So that's what this model tells us. Okay, so that's interesting. Now let's use that in equation 3. When equation 3 has no diffusion term. So equation 3, together with this equation 3, what it means is that you see the product of I and M is basically constant because C is constant. Right? So this is, I'm moving this here. So the product of I and M is constant, is alpha C P C0 divided by K is also constant. That's interesting. That means that where there's high inhibitor, there's low morphogen. Where there's high that makes sense, right? And now let's use that in equation 1 to find the. So in equation 1, we have di times d2i over dx squared equals kim. This is what I got from moving this here. So, and this is constant. This is constant. Kim is constant. And I'm going to, instead of I, I'm going to write, because I times M is constant, instead of I can write, what is I? It's this constant divided by M. So this is, instead of I can write, so this is d2 dx squared times 1 over M times all this constant, alpha C P C0 divided by K. Right? Because I equals this. And that equals alpha C P C0. So this falls out. Nice. That's nice. And I get an interesting thing. The second derivative of 1 over M is just a constant. What function has a constant second derivative? A parabola. The linear term vanishes when I take the second derivative, and the square term, just second derivative, gives me the coefficient. So this is equal. So 1 over M is something like, ax squared plus bx plus epsilon squared. I'm just writing it like that. This is a parabola. That's the only kind of function that second derivative is constant. And because of symmetry, again, there can't be, this term has to be 0. Because otherwise I'll have a parabola that's slanted either this way or that way. You can't have that because of symmetry. And so we end up with a solution. Our solution. Say again? This? I replace I with this thing. So I have 1 over m times this constant. And from this equation, I know that the second derivative of I is equal to this. Okay.
But how did you get the next line? I mean, the di over k?
Oh, I'm sorry. Yeah, so this I divide, and so I get 1 here. And so now I multiply by k.
So it's k over di?
Oh, yeah. Thank you. Thank you. Good question. And by the way, by asking this, you really helped a lot of other people who are thinking of the same thing. And great. That's correct. That's correct. So, so our answer is, I'm going to erase all the equations here because I'm just going to write the answer. The robust models have this following solution. That since 1 over m is a parabola, m is just 1 over a parabola. So this is a kind of interesting profile that, oh, I should say, by the way, I'm sorry, I'm going to define x equals 0 here, is here in the middle. Will be our definition. Okay, x equals 0 is right in the middle. For all the symmetry things I talked about, it's important. X equals 0 will be in the middle, and this will be, x will be plus L, and here x equals minus L. That's our coordinate system. So this kind of function is highest in the middle, and then decays like 1 over x squared as you go away from the middle. So this was important. I forgot to mention. That's important for all these symmetry things I talked about. So 0, everything has to be symmetric around 0. And because of this k over di thing, in fact, A equals to di over k, because this is 1 over m. So, and there's another factor 2 somewhere here. That's just a detail, but. Yeah, 2 di over k. The 2 comes because when you take a second derivative, you have 2a. That's the second derivative here. Okay. And epsilon squared is something like, has to do with the total level of m. So the total level of morphogen you get by integrating all this. And this epsilon, the more morphogen you have, the smaller this epsilon. So let's just look at the solution. It's very interesting. If m tot is large, epsilon of course is small, because epsilon squared goes 1 over m tot. And then m of x looks like, so this is very similar to what we had last time. There can be a region, a small region of size epsilon where changing the morphogen concentration makes a difference. I'm going to try to plot this again. If I change the total level of morphogen, let's say, it makes a difference, but only in a small region of size epsilon. And I can make very, very small if I make the morphogen just high, high, high. And outside of this region, this goes like something that depends only on the diffusion constant and this on rate, and does not depend on all of the other parameters, alpha c, d, c, protein level, I level, mtot, et cetera, et cetera. It doesn't depend on any of the levels of the proteins. That depend on most of the biochemical parameters. So we find that these robust models are a tiny fraction of the possible models. For some reason, they're easy enough to solve analytically. That's something, for some reason, happens again and again, that the robust models are easier to understand than the general model. I don't know why that is. Is there a link between understandability and robustness? Who knows? They give power law tails. So instead of exponential tails, they give these long-range tails. They make two biochemical predictions. And in fact, both of these biochemical predictions turned out to be correct. And especially this one that the morphogen only diffuses when it binds to the inhibitor was tested after the model. The prediction of the model, and turned out to be correct also. So you can find these biochemical predictions. And now we can ask ourselves, how does this work? What's going on here? How does this work? And the name of this mechanism is a shuttling mechanism. The way it works is like this. So the inhibitor is made in the sides, and it diffuses in, and it acts. It has two things. It does. It stops the morphogen from signaling, and also allows the morphogen to diffuse. So that's an interesting two functions in the same molecule. So what's happening here? So it's where there's in regions where there's low morphogen, or I should, the way this works is like this. I binds M, allowing it to move, but this complex leads to I degradation. So M is released and kind of stays put. So when M starts building up here in the middle, I molecules that happen to come here, they bind M, and then they get degraded. So M stays put there. So in regions where there's low I, M can't move, and it gets accumulated. The more it's accumulated, the less I can enter, because when it enters, it binds M, and gets degraded. So there's a kind of a weird kind of feedback loop that the more M is deposited in the midline, the less it can move, because it creates a barrier for the taxi to come in, because it picks up a passenger and gets degraded. So I just want to see how I wrote down a better explanation. So I wrote down the less I there is in the middle, the more M accumulates. And the more M accumulates, the less I there is in the middle. Something like that. Again, it's difficult to think of this without doing this, I think, this parameter scan and finding robust models, but that's That's the way it looks. And it looks like these three molecules are found together. These three, or homologs of these three molecules, similar molecules, are found together in very many different kinds of patterning events in different animals. So, opening, in particular, in the frog egg system, which is very well studied too. So, could be a kind of general mechanism. Who knows? Needs to be tested. What do you think?
Why is it nice to go just to the center and not to the others?
Sorry. It does go. It does diffuse.
Why is it complex? INM is shuttling just to the center and not to the
Why is the complex suddenly just to the center? It's subtle. It shuttles everywhere. It diffuses. The thing is, it gets degraded too. So it diffuses and gets degraded. When it gets degraded, M stays put, finds another inhibitor, starts moving. But if this M by chance comes to the middle, that's special because the inhibitor is lowest there because of just of this diffusion process. And therefore, M has the smallest chance to move there. So it builds up. The more it builds up, the more the likelihood of an I entering there to bind an M and get degraded. So there's this feedback where the more it accumulates in the center, the less taxis can come in to pick it up. And so, in fact, there's something very special about this mechanism. All of M, it accumulates here. In other mechanisms, the total M is constant. It's just that the free M accumulates near the middle. That's just a detail. So, yeah.
Possibly, time will just create very steep graph, like very sharp middle and.
Yeah. So what you're saying is that this mechanism will lead to a very steep graph.
As if x wasn't squared, but
Something,
power of
yeah.
n.
So in fact, that's why we solved the equations. If there's a lot of M, then this looks like this. And this whole process, this whole feedback leads to a power law tail. It's not that M vanishes from here, because I's do enter and can take the M outside too. I mean, it's a diffusion. It doesn't have a direction. So, and then if M is very high indeed, then changes in M only the epsilon is so tiny. The region that's affected by parameter changes is really, really tiny here in the middle. So you can get these long range. Now, it's also not clear to me why both mechanisms we talked about last time and now have 1 over x squared dependence. And both, is that somehow connected intrinsically to robustness of patterning or something? I don't know. But it's very nice to have long range, non-exponential processes.
Can you explain why the M molecule would diffuse only with the taxis? Why not?
Yeah. So the way Nama explained it to me was that she thinks that the thing is that the morphogen actually binds the receptors and doesn't move. And when it's bound to the inhibitor, it exactly blocks the part of M that binds to the receptor, and then it does move. But of course, there could be more complicated explanations. M needs to have a sticky part that binds to something, extracellular matrix or something. So the inhibitor, when it binds to M, it blocks that part. That, yeah. What do you think?
Where do we see the dependence on initial conditions and boundary conditions? Where, for example, the length L?
Mm-hmm. So this solution is good for the region which is far away from L. So if near L there could be corrections. The other boundary conditions really don't matter except for in this epsilon region near this x equals 0. So I guess you can say it's also in a certain way robust to boundary conditions in a sense. But near the place where I is secreted, I think there's differences, but I'm not sure. It could be that this also lasts there. I'm not sure, because M also goes into that region where I is secreted, so it could be. But then there's additional terms in the equations, which are production terms of I. So the equations change in the regions next to those terms. I don't know, I have to think about that.
But will it change the whole picture if I decrease L by 2?
Okay, so the question is, if we take the dorsal region and let's say decrease it by 2. Yeah. So imagine that even when you decrease by 2, it's still far away. It won't change this. And that's actually a problem not for the fruit fly, but for other organisms, because the fruit fly cylinder is pretty uniform from baby to baby, but the frogs and us are very different. You can have babies that are, 1 kilo and 4 kilos, and they are very good proportional bodies, but they're just much less mass. So in frogs, for example, you can take an egg and take a hair from your eyelash and very delicately pinch the egg in half, and that egg develops into a perfect frog that's half the size. So there's that phenomenon called scaling. And that means that if you change the boundary conditions, the entire profile changes. That doesn't happen here in these equations. Like you said, I changed by 50%. I have the same profile. So if I'm not changing the thresholds, I'll have the same pattern, but it won't scale with the total length of the... In 2008, Ahmad did, and Dani Ben-Zvi did a really beautiful study of the frog, experimental theoretical, where they found robustness and scaling. And it's the same thing, same shuttling mechanism, except it has 2 different morphogens together. And one inhibitor that's a shuttle. And I'm debating whether to do that next time, maybe next class I can show you that. Because again, you can solve it on the blackboard, everything. And parameter scan, very small number fraction. Robustness plus scaling is almost like two opposite things. You need to be sensitive to this change in size of the total size, but still robust to changes in the protein levels. So in fact, that's right, like the frontier now, I would say. But so some organisms need scaling, other organisms don't. Higher organisms definitely need scaling. And there must be many more mechanisms to do that. Do you want to know about scaling? Should I do that next time? Who wants me to do that next time? It's about half. As opposed to what, right? So because we have just a limited number of classes left, we have to give something up, right?
Can continue next semester.
Yes. Yeah, before we finish, I want to do something together with you. It's a kind of a teaching thing called dyads, and it's also a way to as a learning tool to help you take together all the information that we learned about in this class. Okay, so the way it works is I'll ask you to turn to somebody sitting next to you and spend a few minutes talking about what we learned today, and especially talk about one thing that you found new and interesting that you didn't know about, or with each other. And then after that, I'll ask some people to just say what it is. And it's a kind of way to summarize the class. Okay, so shall we try it? Yeah? Go into the unknown. It could also be someone behind you if you don't find a partner next to you. Okay, so I'll take a few minutes. I'll kind of clap when it's time to stop. Okay, good. So I'm interested just to name just a few things that were just, so we're going to summarize this lesson, or this is what we learned here. Just some points that you found particularly interesting, so who could just throw out? Who will be the first? Come on. The silence. Yeah, thanks.
Okay. I like the process that Barkai and did. They took some model that they couldn't explain, did the equation that they couldn't solve, and then start randomly search for solutions on the computer. And when they find the solutions randomly, the solution that can answer the question, they did a reverse process to ask, okay, this is the solution. Why did it work? And try to go back to the physics and the biology, and to explain the solution by the.
Okay.
It's a nice process.
Yeah. Something else?
I like the fact that different models have details in common. Like we had in both this class and the previous class model that we got a 1/x squared profile, and another thing is common to the, I think, or maybe common to the chemotaxis robust model, that here degradation depends on its activity, in a sense, its binding, and in that model there was a different mechanism with respect to active and inactive receptors, that only active receptors could get
I see.
demethylated.
Right, in both models there was like some reactions were not allowed or something like that.
Depending on the state of activity of the receptor or molecule.
Yeah, okay. Activity dependent kind of dynamics. Yeah, that's a good point. Yeah.
It's nice also that the simplest model is the one that works, or seems to work. Or one of the simplest.
Or the robust model is somewhat simpler than the general model, generic model. See, it's rare in public to have quiet, right? In public situations.
It's nice that the biochemical predictions were proven.
Yeah. Hmm?
Has the shuttling mechanism been discovered as a result of that study?
Yeah.
Yes?
This is 2004. It was published in 2004. And there's been a whole range of experiments following that to test it in different systems, different groups. And
So the whole mechanism of shuttling was not known before?
no, it wasn't known before that. And these two facts, or these two details were, I think that free I doesn't get degraded. I think that was, there were hints from that from in vitro studies. But the thing that free M doesn't diffuse, only that morphogen only moves when it's bound to inhibitor, was just a surprise, and it turns out to be true, and that opens up this mechanism.
And was it proved by other experiments not related to this one?
Yeah.
The first.
Yes. Okay, so I see our experiments with dyads was led to a lot of silence, but that's okay. I hope you keep talking to each other after class whenever you collide. And let's take a nice deep sigh of relief. Thank you.